Hello class, welcome back. Last period we discussed about the active and bulk transport, its introduction. So in bulk transport, what happens? In this process, the cells takes the food. Okay, the cells take the food or excrete the food in a very large quantity. It does not take in a small amount, but take in a large quantity, bulk. So in the bulk process, you'll find two types, that is endocytosis and exocytosis. What endocytosis means? What endocytosis means? Taking in. Okay, endo means in and exo means out. So in endocytosis, the food are taken in, and in exocytosis, the food are thrown out or excrete out. So in endocytosis, the flexibility of the plasma membrane also enables the cell to engulf food and other materials from its external environment. Such processes are known as endocytosis. It is the active cellular intake of materials. It is of two types, that is phagocytosis, cell eating, and pinocytosis, cell drinking. So in endocytosis, the flexibility of plasma membrane. So what happens? Plasma membrane are made up of two bilayers. Okay, phospholipid, right? So in this process, they allow the food, or the cell are allowed to take the food in a large amount. So this cell engulf the food. Engulf means covered. Okay, like in amoeba, plasmodium, the plasmodium engulf the food, right? So that process is taken in this place. Now let's see the phagocytosis. The transport of solid material like food, pathogens, and foreign matter across the membrane by forming detachable vesicles is called phagocytosis or cell eating. It is a common method of feeding among the protozoans such as amoeba and lower metazoa, example sponges, for engulfing food particles. Now you see the example of amoeba and metazoa, right? So in this process, the food are engulfed, or means the food are covered and they swallow it. They do not have mouth. Okay, they do not have mouth. Plasmodium act as the mouth of this type of cell. So the sponges and amoeba, they take the food directly. In amoeba and other protozoans, the food particles get surrounded by an area of plasma membrane cell surface, which form an invagination along with the food organisms. The invagination pinches off into the vesicles. Such vesicles with food particles are known as phagosomes. So these vesicles, what happens? They act as the mouth, or they act as the hand of the cell. So this type of vesicles are called as phagosomes. So in this amoeba and protozoa, right, the food particles get surrounded by the vesicles, that is phagosomes. So let's see the pinocytosis, that is cell drinking. The intake of droplets of liquid matter, fluid along with submicroscopic particles is called pinocytosis or cell drinking. So in this type they do not take the solid matter. Okay, in phagocytosis they take the solid food particles, but in this pinocytosis they take the water molecules or they take the other liquid molecules. They does not take the solid particles, they take the liquid particles. Because of this they are called as cell drinking. Pinocytosis help in the intake of solids such as insulin and lipoproteins in concentrated form. In this process, extracellular fluid adheres to specific receptors of the membrane and invagination is formed. This is drawn inwards and pinched off as a membrane-bound vesicle, the pinosome, containing the solute. The pinocytic vesicles move into the cytoplasm and its contents are assimilated. Pinocytosis is a common occurrence in the cell lining, the blood capillaries, lungs. Sugars and amino acids enter the cells by pinocytosis. So in this pinocytosis there is a vesicle just like in phagocytosis. In phagocytosis the vesicles are called phagosomes, and in pinocytosis the vesicles are called pinosomes. So these vesicles, they take the liquid material and directly goes into the cytoplasm, where this cytoplasm distributes to different parts of the cell organelles like Golgi bodies, endoplasmic reticulum, right? So these different types of food are assimilated inside the cytoplasm. Now let's say in this pinocytosis they take the materials like insulin. Insulin means like enzymes, okay, enzymes for manufacturing the proteins, lipids, okay. These are the things they take, okay. Ions, sugars or carbohydrates they take in the form of the liquid material. Exocytosis, the process of exudating the secretory materials from the cell is called exocytosis or cell vomiting. You know what is vomiting, right? Throwing out from the mouth. Since the cell does not have anus, right? Since the cell does not have anus like animals, so they take the food as well as they throw the food from their own mouth. Like, because of that it is called cell vomiting and not excretory. Okay, excretory means throwing out from our anus, but in here cell vomiting is mentioned, so only vomiting takes place, not excretion. Okay. Exocytosis occurs in various cells to remove undigested residues of substance brought in by the endocytosis. In the gland cell, secretion collects in a membrane-bound vehicle which now moves to the surface. Here it fuses with plasma membrane. In the fused membrane, a chemical channel, is formed and the contents of vehicle are discharged outside the cell. So these cell vehicles, the undigested food, or say which the endocytosis takes, right? The food which endocytosis takes place, the exocytosis throws out the undigested residues of materials or any materials which or any food which endocytosis takes in. Okay, so this vacuum must be cleared out for other things to come and occur, right? So because of that, everything is thrown out with the help of the exocytosis. So that's about the endocytosis and exocytosis. Now you draw the figure of 1.21 and 1.22 and keep it in your fair copy. That's all. Thank you.